Hi everyone. This is the second of two videos on DNA structure and replication. So what I want to do to start this video is just explain a simple overview of DNA replication, then we'll get into the more specific details. In general, what needs to happen to DNA before it can replicate is that the hydrogen bonds connecting the bases inside the double helix of DNA need to be broken. Once they're broken, then those two strands form what we call the template strands. And enzymes will read the bases that are on each of the two template strands. And as they read those bases, they'll take free nucleotides that are floating around in the environment surrounding the DNA. And they will add those free nucleotides that are complementary to whatever the template is. So if the template's G, those enzymes are going to pull in a C and they'll hydrogen bond those. And then we'll have two daughter DNA molecules at the end. Each daughter molecule will have one side that acted as the template strand and the other side as the newly synthesized strand. All right. So in eukaryotes, the process for DNA replication happens in both directions. And prokaryotes undergo replication a little bit differently than eukaryotes do. We are eukaryotic, so I'm going to start with prokaryotes, and then we'll move on to eukaryotes afterwards. Prokaryotes have about 4.6 million base pairs, compared to humans that have about 3 billion base pairs. So prokaryotes have a fairly small genome, and the time it takes for a prokaryotic genome to be replicated is a lot smaller than it is, a lot shorter than it is for eukaryotes. And another big difference between the DNA that's in prokaryotes and the DNA that's in eukaryotes is that prokaryotic cells have chromosomes that are circular as opposed to linear that are in eukaryotic cells. And prokaryotic cells the DNA only has one origin of replication, and that origin of replication attaches to the plasma membrane that's present in prokaryotic cells. And since they only have one origin of replication, it is going to move in both directions from that one origin of replication. And the replication is going to move, like I said, in both directions, similar to how replication is going to happen in eukaryotes. And so what's going to happen is you're going to start at the origin replication, and these enzymes are going to start breaking the hydrogen bonds in that DNA. And as they break the hydrogen bonds in the DNA, you're going to expose the template strand. Other enzymes are going to come in, grab onto that template strand, read the template strand, and add nucleotides that are complementary to that template strand. So that will form basically two circles that are connected to each other. And once enough nucleotides have been added to each of the template strands, there will be another sequence of DNA that signify or signal to enzymes that replication is about to finish, and you should splice the strands so that the two circles can disconnect from each other. And when that happens, now we have two identical daughter DNA molecules, two identical molecules of DNA. And you can see the animation down in the bottom left-hand corner that describes exactly that process. You have the origin of replication, two replication forks form, and as they form, you're exposing the template strand. That template strand can be used as a template for adding new nucleotides, and then you'll form two identical circles, and then those circles need to be cut from each other in order to separate them. And that's basically how it happens in prokaryotic cells. In eukaryotic cells, like I said before, there's significantly more genetic information compared to in prokaryotic cells. And DNA replication is going to happen in both directions within eukaryotic cells. And it's going to start at a site that we call the origin of replication, just like it started in prokaryotic cells. And in eukaryotic cells, there are multiple origins of replication. We have origin of replication all over our DNA, as opposed to in prokaryotic cells where there's only one origin of replication. And having multiple origins of replication helps because that means that DNA replication can occur simultaneously at many different locations along that super long eukaryotic chromosome. In the example that's shown on the screen, there are three origin of replication. And that means enzymes are going to come in, stop at any of those origin of replication or ORI regions, and they're going to form what we call a replication bubble. And that's basically a bubble where the two template strands are split apart from each other, and then enzymes run in both directions, both to the right and to the left of the origin of replication. And as they do that, the template strand is read, and the newly synthesized nucleotides are added based on that template strand. And one benefit of having multiple origin of replication is that we can have DNA replication going simultaneously, and as the bubbles get larger and larger, they'll start to coincide with each other and form connected bubbles, and you can see that in the GIF that's at the bottom of the screen. So as time is passing, those replication bubbles will increase, and then one replication bubble will link up with another replication bubble, and that larger replication bubble will link up with another one, so that at the end we have two daughter DNA molecules that are formed. And on the next slide, we'll see the linkage of these two replication bubbles as the enzymes move in both directions from the origin of replication that starts in the middle of each of those replication bubbles. And then you'll see at the bottom there is an image of DNA being replicated in a cultured Chinese hamster cell. And the little red arrows that you see inside that micrograph indicate the directions of DNA replication at the two ends of the bubble. It's kind of cool to be able to see the replication forks in that micrograph as they're forming. And you notice how there is, you'll see there are three replication bubbles. Only two of them actually have arrows, but they're all of different sizes. And those different sizes indicate which segment of DNA started replicating first. The one that's the largest would have been the one that replicated first. The second largest would be next, and then the smallest would be the last one to begin, for those enzymes to begin replicating that DNA. So we can use comparisons between size of replication bubble to indicate when that segment of DNA started replicating. Another reason why replication bubbles help in eukaryotic DNA replication is that if we only had one, it would take way too long for replication to occur. And we estimate, I think it's around 5 times 10 to the fourth replication bubbles or origins of replication inside the human chromosome. And given that we have about 23 chromosomes containing about 3 billion base pairs, we need as many replication bubbles as possible to be able to feasibly replicate the DNA in a short time period. And so on this slide, we'll see that DNA replication is happening in a specific direction. And the direction that DNA replication happens in is a 5' to 3' direction. And what does that mean? That means that if enzymes are running on the DNA template strand in a 3' to 5' direction, those enzymes are pulling in free nucleotides, and they're making a daughter strand that starts at the 5' end and ends at the 3' end. So new nucleotides are being added at the 3' end of the nucleotide that preceded it. And that directionality is really important, and I'll explain why it's so important using the next slide. But just know that enzymes that are doing the replication, the enzyme in particular called DNA polymerase, has to work in the 5' 3' direction when it's synthesizing, when it's adding those new nucleotides. So it travels, it moves on the template strand in the 3', 5' direction, but it adds new nucleotides, it synthesizes new nucleotides in the 5', 3' direction. And those numbers in terms of directionality are pretty important. And why are they so important? The reason why they're so important is because of the need for an energy source to power those enzymes. And so given that DNA polymerase has to always attach new nucleotides to the 3' end of the sugar with covalent bonds on the previous nucleotide of that strand, it makes sense that new nucleotides are going to be added in a 5', 3' direction. Look at this image of this molecule. What does that remind you of? It should remind you of ATP, the energy molecule that we discussed when we were talking about energy production. And ATP has 3 inorganic phosphates all bonded to each other at the 5' carbon of the sugar molecule. And those 3 inorganic phosphates are highly unstable. And if we pluck off 2 of those inorganic phosphates, we are able to transfer that energy to power enzymes. And in the process of DNA replication, that ATP molecule, which also serves as the genetic information molecule, we notice that energy that's needed to power the enzymes actually comes from the nucleotide itself. And so the nucleotide that's shown is called an NTP, and the reason why we use N as opposed to ATP or GTP or CTP or TTP is because N just stands for the generic version of the molecule, meaning the base isn't named. The one that we've talked about is ATP, and that's because it's an adenine base that's attached to the sugar. But this applies for guanine bases that are on the sugar, and thymine bases, and cytosine bases as well, and uracil as well. This is the modified version. This is a DNA nucleotide, and so we call it a DATP. This is deoxyribosadenosine triphosphate, and this molecule here is what is going to both be added to the new strand of DNA, but it's also going to power the enzyme that is allowing it to be added, which is a really fascinating connection. So if you look at this diagram here, you'll see that the new strand is forming 5', 3', and in order for that new strand to form, we've got to pluck off 2 of the phosphate groups that are on that adenine as it's being added to the strand, newly synthesized strand of DNA. And then another cytosine is going to come in, and in order for that cytosine to be added, again, in order to power that enzyme, we've got to pluck off those phosphate groups. And then once those phosphate groups get added, then we are able to connect the that side of the DNA molecule with phosphodiester bonds, with covalent bonds. If the enzyme didn't work in that direction, then the enzyme wouldn't be able to power itself. It wouldn't have some energy source. It would need an additional source of energy in order to get that enzyme to work. And that additional source of energy in life doesn't exist. It's coming from ATP. And so the fact that ATP can be an energy source, but also a molecule that is used in the passage of genetic information from parent to offspring, is an awesome trait. So there's going to be the enzyme that's doing this is called DNA polymerase 3. And that enzyme is the polymerase that does the normal base pairing and forms the hydrogen bonds during replication. There's another polymerase that's called DNA polymerase 1, and that DNA polymerase is actually used to replace these RNA nucleotides that are called primers with DNA nucleotides. And we'll talk about what that, why that needs to happen a little bit later. but I just want you to realize that there are two DNA polymerases. One is labeled DNA polymerase 3, another one is DNA polymerase 1. They don't serve the same function. They act similarly, and they both work in the 5' to 3' direction, but they are doing different things at different times. All right, so what we're going to do next is watch a short clip about DNA replication so that you can see in general what's going on.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Now, we heard a couple of different enzymes named in the video. The first one that we heard is an enzyme called helicase, and that enzyme is used to open the double helix up, and it forms these replication forks by breaking the hydrogen bonds that are holding the two strands of DNA together. Hydrogen bonds, or attractive forces between two molecules, could easily, if they're not held apart by something, could easily come back together, and those two strands could come back together, except we have these things called single-strand binding proteins, or SSB proteins, that sit on both of the single strands of DNA as helicase is running and breaking those hydrogen bonds, and their purpose is to prevent the reforming of the helix before that DNA can be used, that single strand of DNA can be used as the template. So it's preventing the DNA from re-annealing, from re-hydrogen bonding together. And because DNA is a helix, as helicase is moving and cutting those hydrogen bonds, the DNA starts to coil up, and it can undergo something called supercoiling if an additional enzyme isn't there to prevent that supercoiling. And supercoiling is because of pressure that occurs on both ends of the replication forks as the DNA is split apart. And it prevents this excessive coiling during replication, and then it helps rejoin the two segments that it just broke or that it just cut to help them be in a more relaxed configuration so that that supercoiling doesn't happen as DNA helicase is running along the DNA, cutting the hydrogen bonds. The way I like to think of it is if you have a piece of yarn and you pull the yarn apart, the two sides of the DNA, two sides of the yarn that are being pulled apart will slowly coil up and potentially break. So this enzyme is helping prevent that breakage in DNA as it's being uncoiled. The other enzyme that you heard was probably DNA polymerase, and that is the enzyme that's responsible for forming the new DNA strand based on the template DNA strand. Another one is DNA primase, and DNA primase is used for synthesizing the primers that are made up of RNA, and that happens both on the lagging strand and on the leading strand of the DNA. And then the last enzyme is DNA ligase, and DNA ligase is responsible for ligating or tying up the fragments of DNA that were formed on the lagging strand. And those fragments are called Okazaki fragments, and they need to be joined up at the 3' end of the DNA fragments to the 5' end of the adjoining DNA. And we'll explain what all these enzymes do coming up. So if you look at the diagram on the top, there is an enzyme called DNA helicase. It starts at the origin of replication, and it moves in both directions from that origin of replication. And as it moves in both directions, it is cutting the DNA molecule at the hydrogen bonds so that they separate and they form this replication fork. And as that's happening, this other enzyme called topoisomerase is moving ahead of it, and we say downstream, in order to prevent the super twisting or supercoiling of the DNA as it unwinds. And then to prevent the DNA from, the single-stranded DNA from reannealing, from rebonding, from rehydrogen bonding, we use these single-stranded binding proteins, or SSBs, to coat and then stabilize the single strands of DNA, preventing them from reforming double-stranded DNA. All right, so like we said, DNA polymerase can only add new nucleotides with a 5', 3' orientation. And before DNA polymerase can do that, there is an enzyme called DNA primase that needs to add a primer to the DNA template strand. And the reason why it does that is because that primer is needed for DNA polymerase to know where to begin. So DNA primase is going to search for a few letters in the DNA template, and as it does that, it's going to add RNA nucleotides that are floating around in the environment around the DNA, and it's going to be, I want to say, about 10 nucleotides long. Of course, could be much longer, but those primers signal to DNA polymerase that you can begin replicating at this segment of the DNA. And DNA polymerase is that enzyme that's going to be responsible for the base pairing of the nucleotides to match the template strand. It facilitates the formation of hydrogen bonds that make up the rungs of the DNA ladder. And it's going to pair adenine with thymine bases and guanine with cytosine bases in the same way that it's going to pair thymine with adenine bases and cytosine with guanine bases. And they're added, those nucleotides are added as triphosphates, with 2 phosphates needing to come off in order to power the enzyme. And those 2 phosphates that come off are called pyrophosphates. They're cleaved off, and then the phosphate is going to be bonded to another nucleotide that's added to form a linked chain that makes up the DNA. Those nucleotides are only going to be added to the 3' end of the newly synthesized strand, and DNA synthesis is going to grow in length in a 5' to 3' direction. But DNA polymerase is going to move or travel in a 3', 5' direction. So there are two strands, two ways in which replication happens. The first way that we're going to discuss is a continuous way of replication. And the reason why it's continuous is because that side of the DNA template strand has the end at the 3' carbon. And since it is at the 3' carbon, DNA polymerase can easily ride on that open 3' end in the 3' 5' direction and synthesize new DNA 5' to 3' direction. And so what you see in this diagram here is that the helicase is splitting and unwinding the two strands of the DNA molecule, and as it moves, the DNA polymerase is going to attach to the template strand, the light yellow strand at the 3' end, and it's going to move 3' to 5'. And as it moves, it's going to pull in nucleotides and make a new strand that's attached to it that's going to be 5' to 3'. And that strand we're going to call the leading strand. And the reason why we call it the leading strand is because it is synthesized continuously in the 5' to 3' direction, as opposed to this other strand that we're going to call the discontinuous strand or the lagging strand, because it starts at the 5' end, and DNA polymerase can't run in the 5', 3' direction. It can only run in the 3', 5' direction and synthesize 5' to 3'. So it's opposite of the way that DNA polymerase can synthesize. And therefore, what's going to need to happen is DNA fragments are going to be synthesized. The DNA is going to be synthesized in fragments, and those fragments are going to be they are going to need to be joined up to form a single strand. And again, what needs to be used in order to make this happen in the discontinuous strand? Helicase. It's going to start at the origin of replication, and it's going to split the DNA and unwind it to form exposed ends. And then DNA polymerase is going to start at the 5' end and move until it hits a thing called a primer. And in this diagram, the primer is labeled in gray. And that primer was added by the enzyme DNA primase. And that primer tells DNA polymerase when it can begin synthesizing. So DNA polymerase is going to attach the 5' end. It's going to travel until it hits a primer. And so it's going to travel upwards until it hits a primer. And as it travels upwards, it's going to hit the primer, and as soon as it hits the primer, now it can synthesize DNA and travel in the 3', 5' direction down this way. So it's going to attach here, move, hit the primer. Now it hits the primer, and it can synthesize 3' to 5' this way. Once it does that, it's going to need to move back until it hits another primer. It hits a primer, and it'll synthesize 3' to 5', moving 3' to 5', and those newly synthesized DNA nucleotides will be added 5' to 3'. And then it will travel upwards again until it hits another primer and do that same thing, and boom, again and again and again, until you have all of these fragments that are about 1,000 to 2,000 base pairs long formed, and then another enzyme will come in and it will attach those fragments together. And the enzyme that's used for attaching those fragments together is known as DNA ligase. And DNA ligase is going to go in and join the neighboring fragments that were separated. One thing that I want to point out is that DNA polymerase can only add to the 3' end of the deoxyribose sugar. And that being the case, synthesis on the lagging strand is opposite of what the leading strand is doing. And the lagging strand must wait until there's enough room in the replication bubble to synthesize in that direction. And when it does, it forms fragments of DNA that are called Okazaki fragments. And as one fragment lengthens and runs into the other, the DNA primers need to be replaced with, the RNA primers need to be replaced with DNA base pairs. And then that DNA ligase enzyme is going to come in and tie together those fragments. And then that strand is going to be called the lagging strand. So this is happening at the same time. This replication is happening in the lagging strand at the same time that it's happening in the leading strand. But the lagging strand is going to always be a little bit behind the leading strand because of the need to move in the opposite direction, in the 5' 3' direction. And that didn't happen in the leading strand. Both DNA polymerase and DNA ligase need to facilitate the formation of covalent or phosphodiester bonds between the backbone of the DNA molecule. And that's pretty important. One way you can remember what DNA ligase does is through a surgical process that you've probably heard of. If a female human wants to get her tubes tied, sometimes we say tubes tied, that medical process is called a tubal ligation. And a tubal ligation is when the fallopian tubes are tied and they prevent the release of eggs into the uterus. So if you assume you tie the end of a tube shut, the egg can't go through that tube. It can't travel through that tube. And so the word ligation or tubal ligation, the surgical procedure, is the same. The word is the same as DNA ligase. DNA ligase is tying together the Okazaki fragments. And so you can see this diagram here that basically goes through the 6-step process of replication that occurs in the lagging strand. And this is just an overview of what's going on with DNA synthesis. In DNA replication, the first thing that needs to happen in the lagging strand is that a primer needs to be added. So we add this short RNA primer using the enzyme primase. As that happens, now DNA polymerase 3 will know where to start, and it can come in the lagging strand and synthesize DNA at the 3' end of the primer, continuing to synthesize in the 5' 3' direction. And as that's happening, another DNA polymerase enzyme called DNA polymerase 1 can go in, remove those primers, and replace those RNA primers with DNA nucleotides. And then the last thing that needs to happen in the lagging strand before that strand is fully synthesized is another enzyme called DNA ligase is going to come in, following DNA polymerase 1, and it's going to ligate together the strands, the Okazaki fragments that were formed. And the DNA ligase is going to covalently bond the sugar molecules together. It's going to form a bond between the backbone. This is a generalized diagram that shows what's happening in DNA synthesis. And what I just want you to remember is that DNA is being replicated in both directions. Enzymes are starting at the origin of replication, at this part here on this strand, and at this part here on this strand. And it is moving outwards to the left and to the right. And this helicase enzyme is zipping along in this direction and zipping along in this direction to break the hydrogen bonds. And as that's happening, other enzymes need to come in and add the primers, for example, in green. Once the primer is added, DNA polymerase can move, boom, down this way and form the red. And on this side, DNA primase comes in, it finds the segment of DNA that it can make an RNA primer at, and it does that, and now DNA polymerase is going to come in, stop when it finds the primer, and then move to add new nucleotides, 5', 3'. And then it's going to travel again, stop when it sees another primer, move, and add new nucleotides, that are 5 prime, 3 prime, move, hits another primer, and go 5 prime, 3 prime, so forth and so on. So now I would like to shift gears a bit and talk about a biotechnology technique that we can use to make copies of DNA and sort of go into the history of how it was developed. The question I have for everyone is, if you wanted to simulate what is going on in the cell to make copies of DNA, what would you need in order to make that happen? And I'll give you one starting material. I'll give you a test tube. You have a micro test tube. What are you going to need to put into that test tube in order to make copies of it? Think of the enzymes that the cell needs. Think of the environment that the cell is in. Think of different things that are needed in order to make a copy of DNA inside a cell. You're going to need all of that stuff in order to make a copy of DNA outside of a cell. So just take a few moments to think about what you might need to put into that micro test tube. The first thing I'd need is the DNA target sequence. What is it that I would like to make a copy of? I can add that to the test tube. Excuse me. And then in order to make a copy of it, I'm also going to need DNA polymerase. I'm going to need the DNTPs, that would be the adenine, the thymine, the guanine, and the cytosine. And they need to be free nucleotides. I'm going to need MgCl2, and MgCl2, when it's in water, dissociates to form Mg magnesium 2 plus ions and chlorine and chloride ions. And why do we need to add that? That's added because the magnesium 2 plus ions act as a cofactor for DNA polymerase. And in order for DNA polymerase to polymerize the nucleotides, it needs magnesium. It comes into the active site and it allows for the phosphate groups to be cleaved off and to bind to those negatively charged phosphate groups. What else will you need? You'll also need primers, and you will need NTPs, so adenine, uracil, cytosine, and guanine that are not deoxygenated, so they would be RNA nucleotides. And you'd also need some sort of pH buffer to maintain the pH of the solution that this is in. All of that stuff would be added to the test tube, and then you would put that stuff into a machine called a thermocycler. And a thermocycler is a machine that can heat up and cool down at specific temperatures at specific times in order to jumpstart the reactions that need to happen. And this technique that we just described is called a polymerase chain reaction. Sometimes we just say PCR. And it's used to amplify segments of DNA that we want to make copies of. So if we want to produce millions of copies of a specific sequence of DNA, and if we are, for example, at a crime scene and we want to make copies of some DNA that we found, we can use this polymerase chain reaction technique to start with even maybe a single copy of that DNA and replicate it millions of times to make multiple copies. Or let's say we have DNA from bone, or we have DNA from a fossil, or DNA from some other substance, or we're able to take very few single copies of that DNA and replicate it. The steps that we need to take mimic the steps that are going on inside the cell. And they need to be controlled. The conditions have to be controlled because this is going on in a lab. And in order for them to be controlled, we have to use enzymes that we can control. And we got the enzymes for being able to do polymerase chain reactions from thermophilic bacteria. You might be asking, why don't we just use enzymes that can come from human cells or from some other cell that's readily available? And the reason why we use thermophilic bacteria enzymes is because those enzymes are from bacteria that live in very hot environments. They're thermophiles, and so that bacteria, the enzymes that are in that bacteria only work at high temperatures. And that's a good thing, because if an enzyme is only active at specific temperatures, and in this case at high temperatures, we have the ability now to be able to basically tell the enzyme when to begin and when to stop working. And that allows us to regulate this process in a very specific way. When we do a lab using polymerase chain reaction in the future, we're going to use a specific enzyme, polymerase enzyme, that's called Taq polymerase. And that enzyme came from a bacteria that's known as Thermus aquaticus. And so they took the first letter and the first two letters of this, first letter of the first word and the second and the two letters of the species. They put them together, and that's the enzyme name. And this Thermus aquaticus only works at a specific temperature, so that we can put the sample with that enzyme, raise the temperature at which that sample is at, and when we raise the temperature, Taq polymerase is activated and it starts working. And when we cool it down, Taq polymerase stops working, because Taq polymerase only works at a high temperature. And that last line, we must use a PCR machine called a thermocycler. In the past, instead of using this thermocycler, this automated this automated machine that regulates temperature, researchers, scientists used to have 3 separate water baths, each of the water baths set at a different temperature, and then take the micro test tube, stick it in the one water bath at one temperature, hold it there for a certain amount of time, pass it to another researcher that's standing in front of the second water bath, stick the micro test tube in that second water bath, wait a certain amount of time, pull it out, hand it to the third researcher, third scientist, and they'd stick it in that third water bath at a different temperature, hold it there for a certain amount of time, and then begin that process again hundreds of times. That process was extremely tedious, took a lot of time, and you needed 3 people in order to do it. A thermocycler allows us to automate that process. It handles the ups and downs of temperature in a very precise way, and allows us to do that without having 3 separate water baths, without having to spend a lot on energy, and it just automates it, so you can do it at any time of the day. Here's basically what's happening. So you take all of these PCR components, put them into the thermocycler, and then the thermocycler will cycle. And one cycle will involve separating the strands by raising the temperature to 95 degrees Celsius. It will then allow for the primers to be added to the DNA in the same way that primers would be added in a cell. It decreases the temperature to 55 degrees Celsius and then increases the temperature again to 72 degrees Celsius. And at that temperature, the enzymes are active and we can synthesize the new strand. So it's a 3-step process. Denature the DNA, that means separate the DNA. Anneal the primers, that means we add the primers. And then the third is the extension stage. Extend the DNA using the DNA polymerase. This video we will watch goes over the basic process of PCR.
Polymerase chain reaction, or PCR, is a simple yet essential way to make copies of a small amount of DNA by exploiting DNA's natural ability to replicate itself when a cell divides. The first step is to extract a small amount of DNA from a sample taken from an individual or piece of evidence. The DNA is mixed with a chemical cocktail including natural enzymes, synthetic chemicals called primers, and 4 nucleotides: adenine, thymine, cytosine, and guanine. After filling tiny plastic tubes with the mixture, a technician places them in a microwave-sized machine that heats and cools the tubes to a series of precise temperatures. The changing temperatures ignite a chain of events, beginning with the denaturing or unzipping of the double-stranded molecules. Next, the primers bind to the individual strands at precise locations, a process known as annealing. Ushered in place by the primers, enzymes steer the
And this is another simulation that I think details exactly what's happening to the DNA molecules as they're going through the polymerase chain reaction. So this one is a little bit different than the first one.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
So now that we talked a bit about the polymerase chain reaction, I want to move back and talk about ways in which DNA can be repaired. Because when enzymes are working, the enzymes initially make a lot of mistakes. And typically DNA polymerase has about 1 mistake in every 10 to the third replications. And that's about 2 errors for every cell division if we have 1 gene equaling about 2,000 bases. That's a lot of errors. And so in order to fix that, cells have 3 basic repair mechanisms for making that happen. And the repair mechanisms bring down that proofreading and error rate to about 1 in a million, which makes it a little bit more manageable. Each of these mechanisms works in a slightly different way. So what we're going to do is go through the different types of repair mechanisms after we talk about ways in which mutations can occur. There are a couple different ways for mutations to happen in addition to enzymes being mutated. The first way is called an intercalcating agent. And an intercalcating agent is a molecule that gets stuck or inserted between bases in DNA, and it alters the shape of the DNA molecule. In order for an intercalcating agent to be removed, the repair a lot of time causes additions and deletions in DNA, and it starts to mess up the message that is coming from that segment of the DNA molecule. There are a couple different examples of intercalcating agents. Proflavin is one, acridine orange, benzopyrene, and then ethidium bromide. A lot of those are staining agents, and we use them to look at different parts of the cell. There's one in particular, ethidium bromide, and ethidium bromide is one that we're going to be using in the lab. And ethidium bromide is used to stain DNA, and we're going to use it in a lab that is called a restriction enzyme and a gel electrophoresis. So we're going to cut DNA and run it on a thing called a gel, and the DNA is going to be stuck inside that gel, and in order for us to see it, we're going to add a dye, and that dye is going to get stuck inside the bases of DNA, and we're going to be able to visualize it. So ethidium bromide does the same thing if we get it on our skin. If it interacts with skin cells, it gets stuck in between the bases of our DNA in the skin cells, and it can alter the shape and eventually affect mutations to build up and cause those cells to not be able to work the way that they're supposed to. Another type of cause of mutation is UV radiation. UV radiation triggers covalent bonds to form between adjacent pyrimidine bases. And UV radiation causes a thing called thymine dimers. And that's when we have 2 pyrimidine bases, 2 thymines, attached to each other, and those 2 thymine bases end up joining to each other. they form a covalent bond between each other instead of hydrogen bonding to the bases that are anti-parallel to it. And that is problematic because those bases jam up the enzymes, the DNA polymerase enzymes that are reading and synthesizing DNA. If you jam it up, it's more likely that that enzyme isn't going to work right, and the fidelity of the message in that gene is going to be lost. And the last way an environmental factor might affect X-rays, affect DNA is X-rays. And X-rays are rays that are part of the electromagnetic spectrum, just like UV radiation is and just like visible light. And X-rays remove electrons from molecules and they generate highly reactive intermediates that damage DNA, something called free radicals. And those free radicals might interact with molecules or enzymes and affect their functionality. So X-rays also play a big role in DNA mutation and can affect how a cell works. One thing that I forgot to mention is one example of an intercalcating agent is benzopyrene. And benzopyrene comes from tar and tobacco, and it gets stuck in the DNA. So if you smoke, or if you vape, or if you joule, or if you use some sort of e-cigarette or something like that, and it contains tobacco, the benzopyrene is altering your DNA because it's intercalcating, it's getting stuck in between the bases of DNA. This image here, this diagram here, shows what ultraviolet radiation does to DNA. So if you look at the two thymine bases here, these two thymine bases will dimerize or form covalent bonds with each other instead of hydrogen bonding with the adenine bases that are antiparallel to it. So these two thymine bases dimerize, and that jams up the DNA polymerase enzyme. Here's a video that describes what happens to your DNA if it's damaged. So we're going to watch that right now.
The DNA in just one of your cells gets damaged tens of thousands of times per day. Multiply that by your body's 100 trillion or so cells, and you've got a quintillion DNA errors every day. And because DNA provides the blueprint for the proteins your cells need to function, damage causes serious problems, such as cancer. The errors come in different forms. Sometimes nucleotides, DNA's building blocks, get damaged. Other times nucleotides get matched up incorrectly, causing mutations. And nicks in one or both strands can interfere with DNA replication, or even cause sections of DNA to get mixed up. Fortunately, your cells have ways of fixing most of these problems, most of the time. These repair pathways all rely on specialized enzymes. Different ones respond to different types of damage. One common error is base mismatches. Each nucleotide contains a base, and during DNA replication, the enzyme DNA polymerase is supposed to bring in the right partner to pair with every base on each template strand, adenine with thymine and guanine with cytosine. But about once every 100,000 additions, it makes a mistake. The enzyme catches most of these right away and cuts off a few nucleotides and replaces them with the correct ones. And just in case it missed a few, a second set of proteins comes behind it to check. If they find a mismatch, they cut out the incorrect nucleotide and replace it. This is called mismatch repair. Together, these two systems reduce the number of base mismatch errors to about 1 in 1 billion. But DNA can get damaged after replication too. Lots of different molecules can cause chemical changes to nucleotides. Some of these come from environmental exposure, like certain compounds in tobacco smoke. But others are molecules that are found in cells naturally, like hydrogen peroxide. Certain chemical changes are so common that they have specific enzymes assigned to reverse the damage. But the cell also has more general repair pathways. If just one base is damaged, it can usually be fixed by a process called base excision repair. One enzyme snips out the damaged base, and other enzymes come in to trim around the site and replace the nucleotides. UV light can cause damage that's a little harder to fix, Sometimes it causes two adjacent nucleotides to stick together, distorting the DNA's double helix shape. Damage like this requires a more complex process called nucleotide excision repair. A team of proteins removes a long strand of 24 or so nucleotides and replaces them with fresh ones. Very high-frequency radiation, like gamma rays and X-rays, cause a different kind of damage. They can actually sever one or both strands of the DNA backbone. Double-strand breaks are the most dangerous. Even one can cause cell death. The two most common pathways for repairing double-strand breaks are called homologous recombination and non-homologous end joining. Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes interlace the damaged and undamaged strands, get them to exchange sequences of nucleotides, and finally fill in the missing gaps to end up with two complete double-stranded segments. Non-homologous end joining, on the other hand, doesn't rely on a template. Instead, a series of proteins trims off a few nucleotides and then fuses the broken ends back together. This process isn't as accurate. It can cause genes to get mixed up or moved around, but it's useful when sister DNA isn't available. Of course, changes to DNA aren't always bad. Beneficial mutations can allow a species to evolve, but most of the time, we want DNA to stay the same. Defects in DNA repair are associated with premature aging and many kinds of cancer. So if you're looking for a fountain of youth, it's already operating in your cells, billions and billions of times a day.
The video talked a bit about different ways in which DNA can repair itself after it's mutated. The first one that I want to go into is DNA mismatch repair. And mismatch repair is when an enzyme can recognize that a base in the DNA is mismatched, is incorrectly matched to its parental strand. So in the example labeled A, you see that a thymine is bonded to a guanine, which shouldn't happen. And that enzyme is going to initiate a repair process. And in order for that repair process to happen, proteins, in this case mut-L and mut-H, are going to be recruited, and mut-H is going to break the backbone of the newly synthesized strand that has the mutant nucleotide in it. And an enzyme is going to remove some of those nucleotides, including the one that is wrong. And then DNA polymerase 1 is going to come in, attach to the DNA, and it's going to use the template strand, that parental template strand, as a way of filling in and replacing the missing nucleotides that were messed up. And then DNA ligase is going to come in and join the backbones, the two strands together. One thing that's interesting about mismatch repair enzymes is that when a mismatch repair, when a mismatch occurs, there is some sort of bulge that occurs, and that bulge is what allows the enzymes that are running to check on fidelity. That bulge indicates to those enzymes that there's a mistake. And there's a high percentage of individuals who are heterozygous for the genes that are coding for those enzymes, coding for the mismatch repair enzymes. And if an individual is heterozygous, that means they have one mutant copy of the gene and then normally one normal copy of the gene. So they're fine most of the time because they still have a working copy of the mismatch repair enzymes. But if a mutation occurs in that one copy that they have, then what we see actually is that there's a high percentage, 46% of the cases of colon cancer that we see are due to mutations in those mismatch repair enzyme genes, in the genes that are coding for those mismatch repair enzymes. And that's pretty scary. There's a big connection between those enzymes and possibly having colon cancer. The other type of repair that I would like to discuss a bit is base excision repair. And base excision repair typically occurs when a uracil is found in the DNA, and that uracil signals to those enzymes that it's not supposed to be there. The uracil is bonded in this case with a G, a guanine. It shouldn't bond, the uracil shouldn't bond to guanine, and an enzyme called uracil glycosylase is going to come in, cleave that uracil from the deoxyribose sugar, and then another enzyme called AP endonuclease is going to remove the sugar, and then an additional enzyme is going to come in, add that missing base, add in the correct base that needs to be replaced. So in this situation, uracil was plucked out, so cytosine needs to come in to match up with the template strand, and the gap's going to have to be connected using some sort of ligase enzyme. So base excision repair is when one base, in this situation normally it's uracil, one base needs to be removed. The base is removed first, and then another enzyme comes in called AP endonuclease. That's going to remove the sugar. Once the base and the sugar are removed, then other enzymes can come in, add the correct nucleotide, and then seal up the nick and repair the DNA. And the last one is called nucleotide excision repair. And nucleotide excision repair occurs when one or more damaged bases need to be repaired. And so an enzyme is going to come in, cleave the DNA backbone at any of the sites, so both to the right and to the left of the area that's damaged. It's going to signal to enzymes to remove that segment, and then DNA polymerase 1 is going to come in and replace those nucleotides with new nucleotides to fix it. One example of a disease that occurs because of dysfunctional nucleotide excision repair enzymes is called xeroderma pigmentosum, sometimes abbreviated just to XP. If we break up that word, xeroderma, xero means dry, derma skin, pigmentosa coloration or pigment. Someone with XP has extreme sensitivity to UV light, and they are typically not able to go outside, and if they do go outside to be in the sunlight, they have to wear lots of sunscreen to protect their skin because of those thymine dimers that form, and thymine dimers are being repaired by nucleotide excision repair. So if you form a thymine dimer, those enzymes can't work properly, and a lot of times they add the wrong nucleotides, and as a consequence, nucleotide excision repair enzymes need to come in and replace them to correct them. And since the person with xeroderma pigmentosum doesn't have functional nucleotide excision repair enzymes, that DNA can't be repaired, and we see a lot of times those people have high skin cancer rates and have a lot of sensitivity to sunlight. And the last thing I want to discuss is the process of aging that happens and the reason why cells age. And the reason why cells age has a lot to do with a certain part of our chromosome that's called a telomere. And a telomere is a small portion of the DNA that is left unreplicated at the end. And so once DNA has been replicated, there's one problem. The usual replication machinery provides no way to complete the 5' end after the RNA primer is removed. So if DNA is replicated repeatedly, that segment of DNA that can't get synthesized once the RNA primer is removed just gets cut off. It gets denatured. It gets cut off, and it actually produces shorter and shorter DNA molecules. So every time the DNA gets copied, the DNA gets shorter and shorter and shorter, because when the RNA primer is removed, DNA polymerase doesn't know where to begin, and it just can't replicate that short segment of DNA. And so to compensate for this repeated shortening process, repetitive sequences of DNA get added, and those sequences are non-coding sequences. They're not responsible for any genes. They're just there to help prevent serious complications and loss of important genes on the DNA in the chromosome. So that they're called telomeres, those non-coding sequences at the end of our DNA in the chromosomes are called telomeres, and they're repeating, non-coding, guanine-rich DNA sequences. And a cell can divide a certain number of times before the telomeres are totally lost. There are no telomeres left. And when that happens, the DNA replicates and it starts to eat away at essential genetic information, and the cell eventually undergoes apoptosis and dies. So what we're going to do is watch this newscast that talks about telomeres and the way that telomeres affect DNA replication, and then an enzyme known as telomerase. It's kind of cool.
And now, eternal youth. Is it in a cage around the corner? News tonight of a breakthrough for some pioneering mice. But we always wonder, what does a fountain of youth for rodents reveal for humans? Here's Sharon Alfonsi reporting.
I feel tremendous.
In the movie Cocoon, it's a swimming pool that turns back the clock for a group of senior citizens. But now researchers have found a way not just to stop, but reverse the aging process. The key is something called a telomere. We all have them. They're the tips or caps of your chromosome, seen here in yellow. This is what it looks like in a young adult. But as you grow older, the telomeres become damaged and frayed, and as they stop working, we start aging, experiencing things like hearing and memory loss. Scientists took mice who were prematurely aged, added an enzyme, and essentially turned their telomeres back on. You can see it before the enzyme, after.
Their brain function improved, their fertility was restored. It was a remarkable reversal of the aging process.
Look at this picture. The mouse on the right has bad skin, gray hair, and is balding, but the one on the left had its telomeres flipped back on.
And you could see that essentially you now have a dark coat color, that the hair is restored, that the coat has a nice healthy sheen to it.
Even more dramatic, the change in brain size. This is before. The mice had 75% of a normal brain, like a patient with severe Alzheimer's. But after the telomeres were reactivated, the brain returns to normal size. As for humans, while it is just one factor, scientists now say by looking at our blood cells and measuring those telomeres, you can get a better idea of how well you'll age. The longer the telomere, the better the chances for a more graceful aging. But as for tinkering with them and turning back our aging process, researchers say we still have a long way to go. Sharon Alfonsi, ABC News, New York.
So like we said, after the RNA primer is removed from the end of the newly synthesized strand, it's not possible to create a double-stranded section in front of this region from which DNA polymerase can then work to replicate the 5' end. And so we use these telomeres, repeated sequences of TTAGG in humans, on one strand of DNA bound to AATCC on the other strand, to compensate for this. And you can think of telomeres like the plastic tips on shoelaces, because the telomeres prevent the chromosome ends from fraying and sticking to each other, which could potentially scramble an organism's genetic information and then could lead to cancer and other diseases and death, etc. And so each time a cell divides, the telomeres get shorter, and when they get too short, that cell no longer can divide and becomes inactive, and it dies. And that process is associated with the aging. And in human blood cells, the length of the telomeres range from anywhere between 8,000 base pairs at birth to 3,000 base pairs as people age, and as low as 1,500 base pairs in elderly people. In comparison, I want you to think that an entire chromosome has about 150 million base pairs. So each time a cell divides, that average person loses anywhere from around 50 to 200 base pairs from the end of their cell's telomeres, which means that cells can only divide about 50 to 70 times before those telomeres get progressively shorter and shorter and shorter and those cells undergo senescence and they die, or the cells undergo severe genetic damage that could cause cancer. A couple exceptions to the rule of telomeres are gametes, germ cells, epithelial cells, and cancer cells, as we'll explain why they're an exception later on. So the reason why they're an exception is because those cells have another enzyme in them. And that enzyme is called telomerase. And in gametes, the shortening of those telomeres would actually cause serious problems. So if chromosomes of gametes become shorter each time during replication, then essential genes would eventually go missing. And an enzyme complex called telomerase catalyzes the lengthening of the telomeres in gametes. And it's called, the enzyme is called a ribonucleoprotein, and it adds DNA sequence repeats to the 3' end of the DNA strands in the telomere region. So it adds TTAGG sequences in all vertebrates at the ends of eukaryotic chromosomes. And this region of repeated nucleotides contains non-coding DNA, and it hinders the loss of important DNA from the chromosome ends. And as a result, each time the chromosome is copied, only 100 to 200 nucleotides are lost, which causes no damage to the organism's DNA. And so we have this telomerase enzyme, which is called a reverse transcriptase enzyme, that carries its own RNA molecule, which is used as a template, and it helps elongate the telomeres, which are shortened after each replication cycle. And this enzyme is also active in germ cells, blood cells, epithelial cells, cancer cells, and it allows them to seem as if they're immortal. And what we see in cancer cells is telomerase is overexpressed. And cancer cells have this strange ability, if they're in a petri dish, of being able to replicate endlessly. As long as you give it nutrients, those cells endlessly replicate. Whereas if we had normal cells that are non-cancerous in a petri dish and we gave it plenty of food, the amount of telomere that those cells have limits the number of replications that the cells can undergo, and eventually they'll die within a couple replications. Whereas cancer cells, that doesn't happen because of this overexpression of the telomerase enzyme that adds the telomeres to the end after each replication goes on. And so telomerase is actually a target for many anti-cancer drugs, and scientists are using that enzyme in the sequence of DNA that is responsible for making that enzyme as a way of figuring out if we can stop cancer, because if we could downregulate or turn off that enzyme in cancer cells, we would eventually be able to cause the telomeres to not continuously be added, and then we would stop the cancer from being able to replicate. And so that ends our discussion of DNA replication and DNA repair and this telomerase enzyme and the use of telomeres. If you have questions, please feel free to stop by and talk.